The main job of the lungs is gas exchange, pulling oxygen into the body and getting rid of carbon dioxide. Normally, during an inhale, the diaphragm contracts to pull downward, and the chest muscles contract to pull open the chest, which both help suck in air like a vacuum. And then during an exhale, the muscles relax, allowing the lungs to spring back to their normal size, which pushes air out. When you breathe in, air flows through the nostrils and enters the nasal cavity, which is lined by cells that release mucus. That mucus is salty, sticky, and has lysozymes, which are enzymes that help kill bacteria. Nose hairs at the entrance of the nasal cavity get coated with that mucus and are able to trap large particles of dust and pollen as well as bacteria, forming tiny clumps of boogers. The nasal cavity is connected to 4 sinuses, which are air-filled spaces inside the bones that surround the nose, called the paranasal sinuses. There's the frontal, ethmoid, sphenoid, and maxillary sinus. Those sinuses help the inspired air to circulate for a bit so it has time to get warm and moist. They also act like tiny echo chambers that help amplify the sound of your voice. And this is why you sound so different when you plug your nose, or if they get clogged with mucus during a cold. So that relatively clean, warm, and moist air goes from the nasal cavity into the pharynx, or throat. The region connecting the two is called the nasopharynx, and the part connecting the pharynx to the oral cavity is called the oropharynx. The soft palate, which is the softer portion of the roof of your mouth behind the hard palate, which is the part you can feel with your tongue, and the pendulum-like uvula hanging at its end move together to form a flap or valve that closes the nasopharynx off when you eat to prevent food from going up into the nasopharynx. Finally, there's the laryngopharynx, the part of the pharynx that's continuous with the larynx, or the voice box. Up to this point, food and air share a common path, but at the top of the larynx sits a spoon-shaped flap of cartilage called the epiglottis, which acts like a lid that seals off the airway when you're eating, so that food can only go one way, down the esophagus and towards the stomach. If anything other than air gets into the larynx, then you have a cough reflex that kicks it right back out. Now, once air makes its way into the larynx, it then continues down the trachea, or windpipe, which splits into 2 main stem bronchi. The point at which they split is called the carina. It then enters the lungs, and the right lung has 3 lobes, the upper lobe, middle lobe, and lower lobe. The left lung just has 2 lobes, the upper lobe and the lower lobe. The right mainstem bronchus is wider and more vertical than the left, which is why if you accidentally inhale something big that can't get coughed up, like a peanut, it's more likely to go into the right lung than the left. The mainstem bronchi then divide into smaller and smaller bronchi. The trachea and the first 3 generations of bronchi are all pretty wide and use cartilage rings for support. Now, taking a look at a cross-sectional chunk of this trachea, there's also a layer of smooth muscle, which has nerves of the autonomic nervous system in it. The autonomic nervous system is made up of 2 basic types of nerves: sympathetic nerves, which are involved in fight or flight mode, like running from a turkey, and parasympathetic nerves, which are involved in the rest and digest mode, like eating ice cream on the beach. Smooth muscle along the trachea and the first few branches of bronchi have beta-2 adrenergic receptors. Going back to that turkey, when you're running, the sympathetic nerves stimulate those beta-2 adrenergic receptors, and this increases the diameter of the airways, allowing more air to get in. But those same airways also have muscarinic receptors, which can get stimulated by parasympathetic nerves, causing a decrease in the diameter of the airways. The large airways are lined mostly by ciliated columnar cells and a handful of goblet cells, which get their name from looking like a wine goblet, and these guys secrete mucus. That mucus helps trap particles, and then the ciliated columnar cells beat rhythmically to move the mucus and any trapped particles from the air towards the pharynx, where they can either be spit out or swallowed. This mechanism is known as the mucociliary escalator. After the first 3 generations of bronchi, though, the airways get more narrow, called bronchioles, meaning little bronchi, and these can stay open without the need for cartilage. Air is conducted through smaller and smaller bronchioles for about 15 to 20 generations, and collectively these are known as conducting bronchioles. These conducting bronchioles receive oxygenated blood from the bronchial arteries. The walls of those conducting bronchioles are also lined by ciliated columnar cells and mucus-secreting goblet cells, as well as a new type of cell called a club cell. These club cells secrete glycosaminoglycans, which protect the bronchiolar epithelium. These guys can also transform into ciliated columnar cells, so they help regenerate and replace damaged cells if needed. Now, all these cells of the bronchioles receive oxygenated blood from the bronchial arteries of the systemic circulation, which is important to keep in mind for later. The last part of the conducting bronchioles are the terminal bronchioles, and then after that, air gets to the respiratory bronchioles, which are unique because they have tiny outpouchings that bud off from their walls. These outpouchings are called alveoli, and there are about 500 million of them in the lungs. Eventually, the respiratory bronchioles end when there's nothing but alveoli. And at that point, the airway is called an alveolar duct, rather than a respiratory bronchiole. And this is the final destination of the inhaled air. The alveolar wall has a completely different structure than the bronchioles. There are no cilia or smooth muscle, and instead the wall is lined by thin epithelial cells called pneumocytes. Most are regular pneumocytes called type 1 pneumocytes, but some, called type 2 pneumocytes, have the ability to secrete a substance called surfactant. Surfactant helps decrease the surface tension within the alveoli and therefore helps keep them open. Just like the club cells, the type 2 pneumocytes are capable of transforming into type 1 pneumocytes, so they can also help regenerate and replace damaged cells. Finally, if a tiny particle ever makes it deep into the lungs, there are alveolar macrophages that can gobble it up and then physically move up to the conducting bronchioles, where they can ride the mucociliary escalator all the way up to the pharynx to be either coughed up or swallowed down. Free from particles, the inhaled air is now in the alveolus, surrounded by mostly type 1 pneumocytes. On the other side of the pneumocytes are endothelial cells that line the capillary walls, which is what holds the blood. This time, though, that blood comes from the pulmonary arteries and is deoxygenated. The pneumocytes and the capillaries are glued together with a protein layer called the basement membrane. So the alveolar wall, the basement membrane, and the capillary wall is really all that separates the air from the blood. And this is called the blood-gas barrier. At this point, carbon dioxide diffuses out from the deoxygenated blood and into the air of the alveoli, which then gets breathed out. And with each breath in, oxygen enters the alveoli and freely diffuses into the blood. That freshly oxygenated blood then heads off to the pulmonary veins, the heart, and then to the body's tissues. Alright, as a quick recap, the respiratory system facilitates gas exchange. Oxygen in the air is inhaled and makes its way through the pharynx, larynx, trachea, large upper airways, conducting bronchioles, respiratory bronchioles, the alveoli, and finally the capillary to be sent to the body's tissue. Then carbon dioxide makes the reverse journey to eventually be exhaled into the world.
Today we're going to continue our discussion of respiratory physiology. We're going to take a closer look at the lungs. As we learned in the previous video, humans have two lungs. The lungs are located in the thoracic cavity, and they are separated from the abdominal cavity by a muscle called the diaphragm. The thoracic wall surrounds the lungs and the heart. The lungs have two membranes that protects them. An inner membrane called the visceral pleura that is attached to the lungs, and an outer membrane called the parietal pleura. Between these two membranes is the intrapleural space that has a little bit of fluid which lubricates the two membranes. The transpulmonary pressure is equal to 4 millimeters of mercury. The pulmonary pressure is 760 millimeters of mercury, which is the same as the atmospheric pressure. However, the intrapleural pressure is slightly less. It's 756 millimeters of mercury, and this slightly less pressure, or as we may say, negative pressure, allows for inspiration to occur. Boyle's law states at constant temperature, the pressure of a gas varies inversely with its volume. So P1V1 is equal to P2V2. What this basically means is that pressure is inversely proportional to volume. So as pressure increases, volume decreases, or vice versa. As volume increases, pressure decreases. So now let's take a look at inspiration. During inspiration, the chest cavity expands. So what happens is that the diaphragm contracts downward and the volume in the chest cavity increases. So according to Boyle's law, as the volume increases, what happens to pressure? Pressure drops. And this is called the negative pressure mechanism, which allows for inspiration to occur. So remember, pressure always goes from high to low. Since the pressure in the intrapleural space drops, it causes the air molecules from the outside, from the environment, to move into the lungs, and hence inspiration occurs. Expiration is the reverse of inspiration. So as the chest relaxes, the volume of the chest cavity decreases. So what happens when volume decreases? According to Boyle's law, since volume and pressure are inversely related, as volume decreases, pressure increases. And what did we say about pressure? Pressure moves from high to low. It always moves from high pressure to low pressure. Hence, if the pressure within the lungs has exceeded the pressure of the atmospheric pressure, then what happens to the air molecules within the lungs? They move out, and expiration occurs.
Hi friends, welcome you all to the debut video of 10 Minute Medico. We will kickstart with the topic neural regulation of respiration. The neural regulation includes the central as well as the peripheral neural regulation. We will proceed with the central regulation first. The centers of respiration are present in the brainstem. There are two centers in the pons, namely the pneumotaxic center in the upper pons and the apneustic center in the lower pons. In the medulla, ventrally lies the ventral group of neurons, and 5 mm posteromedially lies the dorsal group of neurons. The neurons of pneumotaxic center lies in the nucleus parabasalis, the ventral group in the nucleus ambiguus and retroambiguus, and the dorsal group in the nucleus tractus solitarius. Coming to the dorsal group of neurons, the dorsal group of neurons are concerned mainly with inspiration by supplying the diaphragm and the external intercostals. They produce a peculiar signal called the ramp signals. The ramp signals are not sharp signals, rather they are graded signals which increase in intensity for a period of around 2 seconds and stop suddenly for a period of 3 seconds. The slow increase helps in graded contraction of the diaphragm, which causes steady and active inspiration. During the pause period of 3 seconds, there occurs a passive expiration due to the elastic recoil of the lung. Two characters of the ramp can be changed. The first one is the rate of increase of ramp signal. This results in faster breathing, but the depth of inspiration remains the same. The second one is the cutoff point. Earlier cutoff leads to faster breathing, but the depth of inspiration here is very less. Coming to the apneustic center, the basic function is to stimulate the dorsal group of neurons. This postpones the cutoff point of the ramp signals, resulting in increased depth of respiration but lowered respiratory rate. Next is the pneumotaxic center, which basically is an inhibitory center. It inhibits the apneustic center and thereby switching off the ramp signals early. This results in increased respiratory rate but shorter inspiration. Finally, the ventral group of neurons, which are mainly concerned with expiration. They supply expiratory muscles like the abdominal muscles and internal intercostals. They are inactive during normal breathing, but whenever there is an increased respiratory drive, these are activated and hence result in forceful expiration. There is also a theory which suggests that the ventral group contains two different groups of neurons, one concerned with expiration and the other concerned with inspiration. These are the central neural control of respiration. The peripheral system contains receptors in the lungs, such as the stretch receptors, which carry information to the brainstem via the vagus, and they inhibit the dorsal group of neurons. So, whenever there is overinflation of the lung, these receptors are activated and they inhibit the dorsal group of neurons, thereby preventing further stretching of the lung. This is a protective mechanism which prevents the lung from overinflation, and this is called the Herring-Gruber reflex. This reflex acts only when the lungs are inflated about 3 times the normal tidal volume. Hence, it is just a protective reflex and has no role in the regulation of normal respiration. There are other receptors, such as the irritant receptors, which when activated by irritants cause coughing as well as sneezing. There are some other receptors called J receptors or juxtacapillary receptors, which are in close proximity to the pulmonary capillaries. These receptors are activated during pulmonary congestion due to congestive cardiac failure or pulmonary edema, and they cause tachypnea, bradycardia, and hypotension, which together are called the pulmonary chemoreflex. Let us also know a little about the effect of lesions of various centers. Consider this graph to be the normal respiratory rhythm. When there is a lesion above the pons, there is no net effect on the rhythm even when the vagae are damaged. But lesion between the pneumotaxic center and apneustic center will cause increased depth of inspiration because the termination of inspiration here depends only on the stretch information carried by the vagus. When the vagus is also transected, there is no feedback for ending inspiration, and this leads to prolonged inspiratory spasm, which is called apneusis. When there is a damage between the pons and the medulla, the control of rhythm by the pneumotaxic and apneustic centers are lost, and this results in irregular breathing, irrespective of the state of the vagus nerve. When there is a damage below the medulla, the total output from the centers are lost, and this results in total apnea.
Spirometry is the process of measuring volumes of air moving into and out of the lungs. The machine used to measure lung volumes is called a spirometer, and the graph of lung volumes created by a spirometer is called a spirogram. The volume of air during normal quiet breathing is called tidal volume. The maximum amount of air that can forcibly be inhaled is called the inspiratory reserve volume. And the maximum amount of air that can be exhaled is called the expiratory reserve volume. Tidal volume plus inspiratory reserve volume plus expiratory reserve volume equals vital capacity. The volume of air that remains in the lungs after forced maximum expiration is called residual volume. And finally, the total amount of air that the lungs can hold is called total lung capacity. To recap lung volumes, tidal volume is the volume of air during relaxed, quiet breathing. The inspiratory reserve volume is the maximum volume of air that can be inhaled after a normal breath. The expiratory reserve volume is the maximum volume of air that can be exhaled after a normal breath. Residual volume is the volume of air remaining in the lungs after forced maximum expiration. Vital capacity equals tidal volume plus inspiratory reserve volume plus expiratory reserve volume. And finally, total lung capacity equals vital capacity plus residual volume.
Whether you're racing in a triathlon or doing something less strenuous, you need to breathe in oxygen to help you get energy, and breathe out carbon dioxide, a waste product. When you inhale, your diaphragm and rib muscles contract, increasing the volume of your lungs. When you exhale, these muscles relax, decreasing the volume of the lungs. When the lungs expand, air pressure in the lungs drops, causing air to flow into the lungs. When lung volume decreases, air pressure increases, causing air to flow out of the lungs. Air enters the nose or mouth, moves down the trachea, and goes into the two bronchi. Air moves down smaller and smaller bronchioles until it reaches a tiny sac, an alveolus. Each alveolus is surrounded by capillaries. Oxygen diffuses from the alveolus to the blood, and carbon dioxide diffuses from the blood to the alveolus. As blood flows through the capillary, it becomes rich in oxygen. In the blood, oxygen diffuses into a red blood cell and binds to hemoglobin, a protein made up of 4 subunits. One oxygen molecule can bind to each subunit. Oxygen-rich blood flows from the lungs to the heart. Which pumps this blood to capillaries all over the body. Here, we see oxygen diffusing from a capillary's red blood cells into a muscle cell. Oxygen is used by the cell's mitochondria to produce ATP during cellular respiration. Carbon dioxide is released. How does carbon dioxide leave the body? Carbon dioxide diffuses from cells into capillaries. Some carbon dioxide stays in the plasma, the liquid part of the blood. Most carbon dioxide, however, enters red blood cells. Some carbon dioxide binds to hemoglobin. The rest is converted to bicarbonate, which diffuses into the plasma. This oxygen-poor blood flows back to the heart, which pumps it to the lungs. There, carbon dioxide diffuses from the plasma into the alveolus. Bicarbonate enters red blood cells and is converted back to carbon dioxide. Carbon dioxide is also released from hemoglobin. Carbon dioxide diffuses out of the red blood cells, into the plasma, and into the alveolus. When you exhale, air flows out of your lungs. And that's how you release carbon dioxide, get oxygen, and keep on running.